thank you so much for the kind introduction. Indeed, it's a great honor to receive this award in my home country. And I'm very excited to present to you today part of my PhD journey at the Netherlands Cancer Institute, where we stumbled upon quite an unexpected mechanism of action of PARP inhibitors. And I would just like to start off by mentioning that PARP inhibitors are nowadays used in the clinic for BRCA1 mutated tumors. These mutations generally occur as germline mutations in women, and unfortunately, they give a very high predisposition to breast and ovarian cancer. Once these women present in the clinic, they're usually treated with a standard of care combined with surgery, chemotherapy, but more recently, also PARP inhibitors have been added to this schedule. And as Johannes already introduced, they are actually based on a very basic biological concept called synthetic lethality. So how this works is that PARP inhibitors do not just inhibit the enzymatic activity of PARP1, but they also trap the PARP1 protein onto the DNA, which generates an obstacle for the ongoing replication fork, and this generates DNA double-strand breaks. In the presence of BRCA1, this is really not a problem. Cells can easily repair this kind of damage and the cell survives. However, in the absence of BRCA1, this results in the accumulation of DNA double-strand breaks and ultimately cell death. Unfortunately, we know that these tumors are very genomically unstable and unfortunately frequently develop resistance through a variety of mechanisms, but the one I would like to focus on for today is the additional loss of 53BP1, and this leads to partial survival of these cells again. So you might agree with me that it's very important to understand the mechanism of action of PARP inhibitors in greater detail in order to tackle resistance. And what we as a DNA repair field often tend to forget is that actually PARP inhibitors do not just work on the DNA level, but of course all of this happens in a chromatinized environment. And in this project, since there's little known about the effect of PARP inhibitors on the chromatin environment, we decided to study this in this project using a variety of techniques. And the one I would like to focus on for today is we performed subcellular fractionation experiments. So we treated cells with olaparib, which was the first approved PARP inhibitors in the clinic, and then performed subcellular fractionation. And as you can appreciate, in this Western blot, there was no difference in histone H3, H4 levels in the nuclei, in the chromatin, or in the chromatin fraction. However, when we looked at the nuclear soluble fraction, we observed something quite interesting. There was this consistent increase in H3 and H4 levels upon olaparib treatment. Further, we could validate this by mass spec. We could also observe the same thing. And actually, we could also observe a concomitant decrease in the chromatin fraction that actually suggested to us that these histones might be released from the chromatin into the nuclear soluble fraction. And this is indeed the case because if we look at the post-translational modifications that these soluble histones carry, they carry mainly post-nucleosomal marks, meaning they have been in contact with the chromatin before. So this was quite exciting, and at this point we really had two burning questions. What are actually the factors that regulate this histone release and turnover upon PARP inhibitor treatment? And can we actually target this to sensitize cells to PARP inhibitor treatment? And to do this, we performed genetic screens. Here we use gene trap mutagenesis screens, but it's essentially similar to a CRISPR screen. So we generated a pool of knockout cells, passaged them in the presence or absence of olaparib for 14 days, and then isolated genomic DNA. And these are the kind of plots that we then get. So genes where the loss causes PARP inhibitor sensitivity will be scored in the bottom of the graph. And these are the results that we got. So in dark blue, you can see all the genes that are differentially regulated compared to the wild-type untreated setting. And of course, we identified many DNA repair genes, but we were also quite excited to find this protein called NASP, which has actually been shown to be a histone chaperone. So looking at the function of NASP, NASP actually exists in two isoforms within the cell, and this mainly consists of TPR repeats which bind histones at different affinities. And its canonical role, once you have synthesis of mu, histone H3, H4, it's shuttled into the nucleus. NASP serves as a storage factor to keep these histones soluble and stable until they are needed by the ongoing replication fork. So in order to see whether NASP was also important for our PARP inhibitor-dependent histone release, we turned back to our subcellular fractionations. And as you can see here, again, in the nuclei fraction, nothing changed. However, when we looked at the soluble fraction, we again, in the wild-type cells, saw a nice increase in H3 levels. but this was completely absent when we knock out NASP, and we could readily rescue this by overexpressing the small S-NASP isoform, actually suggesting to us that also for PARP inhibitor treatment, NASP is important to keep these histones stable. But what happens when NASP is not there? So there's been different routes described in literature that degrade histones, and here we just used two examples, MG132 and baphyllomycin. And as you can see here from this Western blot, we could readily rescue histone stability by treating cells with MG132, suggesting to us that in the absence of NASP, these histones are degraded by the proteasome. So this is of course nice, but we were also wondering if this actually affects viability of these cells and olaparib sensitivity. So we exposed a variety of cell lines that are NASP knockouts to olaparib. And as you can appreciate here, these cells were indeed nicely sensitized to PARP inhibitors. This experiment we performed in mouse mammary tumor cell lines, which are BRCA1, P53, 53BP1 deficient. And the nice thing about these cell lines is that we can actually transplant them back into the mouse mammary fat pad of animals, and then upon tumor outgrowth, treat these animals with either vehicle or olaparib. And as you can appreciate from this survival graph, of course, parental tumors are resistant to PARP inhibitors. They did not respond and resulted in short survival of the animals. However, you can see in the blue dashed line that the NASP knockout tumors responded to treatment, leading to longer survival of these animals. So this was very nice to see. Given that we observed PARP inhibitor sensitivity, we were also wondering whether we could see signs of genomic instability in these cells. So we performed a DNA fiber assay where we pulse cells with for a certain amount of time with different nucleotide analogs. And I hope you can already appreciate from these pictures that loss of NASP resulted in reduced replication fork speed, and this coincided with increased levels of gamma H2X, the induction of DNA damage. Until now, I've shown you a couple of examples how histone turnover might affect viability, but to directly prove that it is indeed the histone turnover that affects these phenotypes and not NASP having a different function, we performed the following experiment. So we turned back to our cell viability, and if we overexpressed histone H3 wild-type levels, this of course does not rescue viability of NASP knockout cells because these histones are degraded by the proteasome. However, we can do a little trick. So there's different mutants in H3 that's been proposed to be targeted less efficiently by the proteasome, and we were actually quite happy to see that these were able to partially rescue our viability. And this allows us to directly link histone stability and turnover to PARP inhibitor response. But as you might see in this graph, there is still a question mark remaining in my schematic. And this is because NASP itself actually does not go to the chromatin. So there need to be additional factors involved in this pathway. And to identify these, we turned back to genetic screens. So again, we took a pool of knockout cells, treated them with olaparib, and then we did not use survival as a readout, but we actually performed a phenotypic screen using NASP levels as a readout, because we reasoned that NASP would be a nice proxy for the abundance of soluble histone in the cells. So we then stained these cells with NASP antibody and sorted the highest and lowest 5%, and then again submitted these samples to next generation sequencing. And these are the results. So of course we see NASP in the bottom as a positive control that are antibody specific, but quite intriguingly, we actually observed many members of the 80 complex. And this is, of course, a very well-known chromatin remodeling complex that is able to not just slide nucleosomes, but also actually evict them. So, and this is indeed the case. So if we knock out one of these components, we can again see that actually these histones do not accumulate in the soluble fraction. And this brings me already to my model. So what we think is happening that upon PARP inhibitor treatment, you have release of these histones by the ANO80 complex. They're shuttled onto NASP. NASP keeps them soluble and stores them until they can be reincorporated into the chromatin by PARP1 and other histone chaperones, which I was not able to show you the data, but happy to discuss further. So I think here PARP1 release actually serves as a double-edged sword. What happens in the absence of NASP? These histones are still released, but they are rapidly degraded by the proteasome, resulting in increased exposure of DNA, the accumulation of DNA double-strand breaks, and ultimately cell death. And with this, I would like to acknowledge, of course, the whole Jonkers lab, especially Jos, for being such a great mentor throughout all these years. Many lab members that contributed. I would like to specifically point out Joseph and Lizzie. Names might sound familiar to some of you. They have been two incredible master students who helped me start off this project. Abdel for all his help with the genetic screens and the mechanistic discussions. Of course, all our other collaborators and funding agencies, and you for your attention. And I'm very much looking forward to your feedback and questions. Thank you.